welcome to the Cognitive Science Colloquium talk. Extremely excited to have you guys here. This is the new Cognitive Science department in the physical space. So the talk will just be here, and then the reception is going to be just down the hall that way. So just exit the doors and go straight, and it'll be on the right. And Caroline Robinson is going to introduce Kalanit.
Great. Thank you all for coming. It's a great pleasure to introduce Dr. Kalanit Grill-Spector. Kalanit, as you all know, is joining us from Stanford, where she's a professor of psychology and directs the Vision and Perception Neuroscience Lab. Just for a bit of background about her, Kalanit actually, I guess probably like many great neuroscientists, started out as a computer scientist, an engineer. Her first degrees are in electrical engineering and computer science from Weizmann, after which she saw the light and pursued a PhD in neurobiology at the Weizmann Institute with Rafi Malik, and a postdoc at MIT with Nancy Kanwisher. My understanding is that she and Rafi Malik developed the first fMRI system in Israel in the 1990s, and which they used to investigate computational vision questions in the human brain. And at this early point in her career, Kalanit became well known for developing the technique of fMRI adaptation, which I literally just taught about today in my Cog Neuro course. So it's very exciting to have a cornerstone member of our field here giving a talk. Her current work is, which we'll hear about today, is really very unique in the field. It sits at the intersection of fMRI work and cytoarchitecture and neurodevelopment. And I'd like you all to join me in welcoming Kalanit.
Thank you, Caroline, for a very kind introduction, and thank you, Jonathan, for inviting me. So it's great to be here. Let me just put this. Okay, so the work that I'm going to be presenting today looks really at the neurodevelopmental aspects of the ventral temporal cortex, and has been done by a lot of students and postdocs in my lab. And in particular, I'd like to highlight the contribution of Jesse Gomez, who did a lot of the work that I'm going to be presenting today. Jesse actually did his undergraduate here at Dartmouth, and he was a Brad Duchesne's student as an undergrad, so thanks for training him. And this is a very exciting week for Jesse because he just started his real position as an assistant professor at Princeton University, so it's been a long way since being an undergraduate here for him. Okay, so recognizing faces and reading words are really important for our daily lives, and we're really good at doing face recognition and reading. We can recognize words and faces in a fraction of a second, despite large variability in their appearance. However, face recognition and reading is an ability that develops across our lifespan. For example, if you look at reading accuracy, children get much better at reading from the age of 5 to the age of 15. And likewise, face recognition ability is much better in teens and young adults as compared to young children. So I'm interested in understanding what are the brain mechanisms that underlie our ability to recognize stimuli, such as faces and words, and also how these mechanisms develop across childhood. So to study visual recognition, we study a processing stream that's called the ventral processing stream. And basically, when you look at objects in the world, light from the objects gets reflected on your eyes, and an image of that object falls on your retina. That image gets transmitted through the optical radiation to the LGN, and from the LGN to the calcarine sulcus, where primary visual cortex, or V1, resides. And then through a hierarchical set of processing areas, including retinotopic areas V1, V2, V3, V4, information gets to this cortical expanse that I'm going to call ventral temporal cortex and VTC, and then you have a percep emerging. Maybe you can recognize that this is Michael Phelps. And I'm going to focus most of my talks on this ventral aspect of the temporal lobe, and it covers an anatomical expanse spanning from the collateral sulcus on the medial side to the fusiform gyrus to the occipital temporal sulcus on the lateral side. So even though it's a well-defined cortical expanse, it's not a homogeneous area, and in addition to having multiple retinotopic regions, it also contains multiple category selective regions. And we identify category selective regions in each individual brain, so I'm going to show you only individual data today. I'll never show you any group map today. And we localize them by putting people in a scanner and showing them stimuli from various domains. So in our experiments, we have faces, bodies, objects, places, and characters. And each domain is composed of 2 categories. So we have adult and child faces. We have bodies and limbs. We have musical instruments such as string instruments and cars. We have objects. And then we have places, houses, and corridors. And we have characters, including words and numbers. And we've spent a lot of time calibrating the stimulus set, so we've controlled it across categories for low-level properties such as contrast, luminance, and pixel-wise similarity. Also, we've controlled it for high-level cognitive factors such as familiarities. So typically in our experiments, we collect 3 runs of these, what we call it the category localizer experiment. And each run has different stimuli, and each stimulus is blocked in mini blocks of 4 seconds each, in which they see 8 images. And this experiment is freely available in our lab's GitHub repository, so if you're interested in using this, feel free to download it and use it. So suppose you're interested in identifying word-selective regions in the brain. What you're going to do is you're going to find which voxels in the brain respond more strongly to words compared to other categories. And when you run this kind of contrast, you will typically find 2 clusters, especially in the left hemisphere, that show higher responses to words compared to other stimuli. People usually refer to this as a visual word form area. The way we like to refer to regions in my lab is by their anatomical location and their selectivity. So I'm going to refer to this as a posterior OTS word patch and the middle OTS word patch. And they're always within this anatomical location, which is this occipital temporal sulcus that I've highlighted. If you look for regions that respond more strongly to faces compared to other stimuli, you typically find bilaterally 2 face patches, one in the posterior fusiform, I'm going to call it P-fused faces, and one more anterior that I'm going to call it M-fused faces. And these regions are always in the lateral fusiform. And if you look for regions that respond more strongly to places like corridors and houses compared to other stimuli, you'll find a cluster of activation that is on the intersection between the collateral sulcus and the anterior lingual sulcus, and this really predicts the location of this region. So what's really interesting about these category selective regions that you can identify them in individual brains, even in young participants. So this slide shows 3 left hemispheres, a ventral view of 6 left hemispheres of different participants ranging from the age of 6 to 22-year-olds. So first of all, you notice that we can find this category selective region in each individual. And second of all, you might notice that they have the same lateral to medial organization. So in all our participants, the place selective activation is on the collateral sulcus. More lateral on the fusiform, you'll find in every participant the face selective activations, and most lateral on the OTS, you find this word selective activation. And I find this topography of category selectivity really intriguing because it raises a very important developmental question. And that question is, why do these regions develop in a similar anatomical location across people? So one clue to this, to answer this question comes from some work from Rafi Malik's lab. So Rafi Malik and his students at that time, Uri Hasson and Ifat Levi, did the following experiments. They showed different kind of stimuli, either presented centrally, indicated here in blue, or peripherally. And then they just contrasted responses to the stimuli, looking for regions that responded more to central than peripheral stimuli. And they found that regions in lateral VTC have a foveal bias, meaning they respond more strongly to foveal than peripheral stimuli. However, regions in medial VTC, shown here in green, had the opposite bias in that they responded more strongly to peripherally presented stimuli to centrally presented stimuli. And interestingly, they also reported a coupling between where these category selective regions reside and these eccentricity biases. Specifically, they found that face-selective regions, shown here in yellow, and word-selective regions, shown here in purple, overlap representations that have a foveal bias, whereas if we look at responses to places and buildings, shown here in blue, they overlap regions of VTC that have a peripheral bias. And basically, this coupling between the location of category-selective regions and eccentricity bias led them to hypothesize that this coupling has to do with the way that we naturally view the world. And basically, they hypothesize that in order for you to recognize faces, you have to use your best vision, which is your foveal vision. Therefore, when you look at faces, you tend to fixate on people's faces. And this consistent coupling between looking at faces and foveal vision is going to generate face representation upon pre-existing, maybe regions in VTC that have this foveal bias. In contrast, in the real world, scenes are big. They will always fill the visual field. So suppose you're sitting in an auditorium like this. It doesn't really matter where you're going to be fixating. There's going to be a portion of the scene that's going to always extend into your periphery. So basically, a place representation for this reason will develop upon peripheral representations of the visual field. So they call the theory eccentricity bias theory, and the theory states that category-selective regions will develop upon cortical representation in which that eccentricity is most often viewed with. So faces will develop on regions that have foveal bias and places on regions that have a peripheral bias. So what I wanted to show you today is several experiments in which we actually experimentally tested the prediction of eccentricity bias theory. So the first question we ask is whether eccentricity bias and visual field representation more broadly develop from childhood to adulthood. So in order to address this question, we performed a population receptive field mapping experiment in 25 children between the ages of 5 and 12 and 25 young adults. And we chose this age range of children because this is an age of dynamic learning where both visual high-level representation, for example, of faces and face recognition ability, as well as reading ability, prominently develop in this age range. So in this experiment, we have our participants lie in an fMRI scanner and ask them to fixate on a central stimulus. Occasionally, the central stimulus changes color, and they're asked to press a button. And as our subjects are doing this fixation task, a bar filled with flickering checkerboards systematically sweeps the visual field. So this lets us map visual field representation in each voxel in the brain. In order to ensure that our subjects are fixating during this experiment, we have an eye tracker inside the scanner, and we can verify that both our adults and children are keeping their eyes glued on the fixation point. So each subject sees 4 runs of this experiment. So we have about 20 minutes of data from the pRF mapping experiment. And in addition, they participate in another scan in which we map category selective regions to look at their eccentricity biases. Okay. So what is a population receptive field? I'm going to call it a pRF in short. So a pRF is a region in the visual space that is processed by the populations of neurons in the voxel. So basically, we're in visual space that voxel process visual information. And we use a computational model to model the pRF of each voxel in the ventral stream. And that computational model is that the pRF of a voxel is basically a two-dimensional Gaussian that has a center, x and y, and a size, sigma. And in addition, it has a compressive nonlinearity that's determined by this exponent, and that's usually less than 1. And this gives us some invariance within the receptive field. So basically, given the stimulus sequence, because we know at every time point where we present visual stimulus, and given the fMRI response of that voxel, we fit with optimization algorithm is the pRF that best explains the voxel response. And what I'm going to show you today are several properties of the pRFs. For example, using the centers of the pRF, we can generate the standard retinotopic maps of polar angle and eccentricity. And in addition, we're interested in seeing the size of the pRFs across what region of visual space each voxels and processes spatial information. So we're going to start from early visual areas because not much is known about retinotopic representation in children. So here I'm going to show you the medial view of the brain, and I'm going to zoom around the calcarine sulcus because this is where early visual areas reside. So this is a zoomed medial view of a child's brain, and this is a zoomed medial view of an adult's brain, and this is a calcarine sulcus. So in adults, we know that V1 has a hemifield representation, so it goes from the lower visual field through the horizontal to the upper visual field, and we can see the same kind of map in the child's V1 in the same anatomical location around the calcarine. In addition, we see that in adults we have several mirror reversals of the polar angle map. And again, qualitatively, we see the same number of reversals in the same location in the child brain as in the adult brain, as I'm showing you for these example participants. We can also use the pRF centers to produce an eccentricity map. And again, you can see in adults we have two eccentricity maps. One eccentricity map in the occipital lobe, so the posterior part of the occipital lobe represents the fovea, and as you move anteriorly, you're going from fovea systematically to the periphery, and you can see the same organization in the child's brain, going from the occipital pole more anteriorly, you're sweeping from fovea to periphery. And in addition, you have another eccentricity map on the ventral aspect of the temporal lobe, going from fovea on the lateral aspect to periphery on the medial aspect. And you can see the same kind of qualitative organization in a child from fovea to periphery in the temporal lobe. So qualitatively, we find the same kind of retinotopic maps in children and adults, and we can use this to define all the retinotopic areas of the ventral stream. But what about quantitative properties of pRFs in these areas? Do these develop between children and adults? So I'm going to first start with the early and intermediate retinotopic areas of the ventral stream, and we're going to quantify several properties of the pRFs. So the first property that I'm going to quantify is the average pRF size in a region. The children are shown in lighter colors, and the adults are shown in darker colors. So you can see that in both age groups, we have a systematic increase of the mean pRF size as you ascend the ventral visual hierarchy, and this is consistent with this hierarchical organization of the ventral stream. Interestingly, we see no quantitative differences in the mean pRF size across children and adults, so also quantitatively, we don't find differences in these regions across age groups. Another thing that we looked at is the mean pRF eccentricity across children and adults. And you can see that even as there are differences across area and the mean pRF eccentricity, again, there are no significant differences across children and adults in the pRF properties in their early visual areas. So one of the things that we like to look at mean statistics, but this is not the only metric that we might be interested on. And another metric that we're interested in is understanding how pRF of a region tile or covers the visual field. So for example, if we take a region like V1, we basically plop all the pRFs that we measure from V1 on the visual field, and we generate what we call a visual field coverage map of an area. So this picture shows you the visual field coverage map. It's the average map in adults of left V1. So you can see that the color map shows you the density of the coverage, and you can see that the redder it is, that you have higher coverage. And as we expect, left V1 in an adult has a uniform coverage of the right or contralateral visual field. And we'll see the mirror representation in the right hemisphere. So the question is, do the pRFs in children also tile the visual field the same way as adults? So this is the data that I showed you for the adults, and this is the average data across our child participant, and we can see that there is no difference in the ways that pRFs cover V1 across children and adults, and this is true for all the early and intermediate retinotopic areas across the ventral stream, that in each visual area we get a really nice coverage of the contralateral visual field in each of these retinotopic areas. So far I've shown you that both qualitatively and quantitatively the pRF properties and visual field coverage in early and mid retinotopic regions of the ventral seam seem to be developed by the age of 5, which is the age of our younger age groups. But what happens in these higher level areas that we and other groups have shown to continue to develop throughout childhood? So I'm going to show you now the visual field coverage from 2 areas. One is this word selective regions in the posterior OTS, and another is from the face selective region in the posterior fusiform or p-fuse faces. And I'm going to show you data from these regions because these regions are still modulated by the checkerboard stimuli, as the checkerboard doesn't really drive the more anterior word and face selective regions. Another thing that you can notice from this example participant is that the word selective region seems to be larger in the left hemisphere than the right hemisphere, and the face selective regions tend to be a little bit larger in the right hemisphere than the left hemisphere. And this is well documented in the literature that there's lateralization, left lateralization for word processing and right lateralization for face processing. In addition, there's an accumulating body from neuropsychological studies like lesion studies, as well as electrical stimulation studies that suggest that the left hemisphere is dominant for word processing and reading, and the right hemisphere is dominant for face processing. So what I'm going to start is showing you the visual field coverage in the dominant hemisphere for each category. So I'm going to show you the visual field coverage in the right hemisphere face selective region and the left hemisphere word selective region. So this is a visual field coverage in the child's right p-fuse faces. So now this is the right hemisphere, so we have coverage of the left visual field, which is the contralateral hemisphere. But if you notice, this visual field coverage is really different than the early and intermediate retinotopic areas because it's no longer uniform. And you see this foveal bias kind of predicted by Rafi Malik's research is that there's higher coverage of the visual field close to the center of gaze compared to the periphery. Another thing that you might note is that this actually doesn't really cover even uniformly the upper and lower visual field, that there's more coverage in the lower visual field compared to the upper visual field. So what happens between childhood to adulthood? So it turns out that between childhood and adulthood, we have a profound development of the visual field coverage in this right face selective regions. First of all, you can see that the foveal bias substantially increases. As you can see, this much more denser hotspot close to the fovea, so it's just as the pRF have moved closer to the fovea. In addition, you can see that there's actually more coverage of the visual field across development, where it extends to the upper visual field as well as the ipsilateral visual field. So we have this profound development in this high-level visual area of the visual field coverage from childhood to adulthood. We can quantify this development by measuring the center of mass of the visual field coverage and seeing if it substantially changes across development. So basically, we measured how far the center of mass of the visual field coverage is compared to the fovea, and you can see that it becomes more foveal or closer to 0 between childhood to adulthood in the right hemisphere, which is a dominant hemisphere for face recognition. Interestingly, if you look at the opposite hemispheres, the left hemisphere, we also have a face selective regions, but we don't see the same development. In fact, we don't see this development at all, and we actually do not see this increase in the foveal bias in the non-dominant hemisphere for face recognition. Okay, so this is already different from early visual areas. What happens in the word selective region? Does it also show changes during development? So in the word selective region, I'm going to look at the left hemisphere because that is the hemisphere that's dominant for reading. And here, because it's the left hemisphere, again, we can see that like earlier regions, we have this contralateral visual field coverage or coverage of the right visual field. And again, a little bit like the face selective regions, we actually see more denser coverage of the lower visual field and the upper visual field. And actually, this doesn't really look like it has a whole lot of foveal bias. But if we look what happens between childhood to adulthood, again, we see this profound shift in the visual field coverage where the foveal bias increases again from childhood to adulthood. And in addition, you can see that the visual field coverage becomes more centered around the horizontal and expands to both the ipsilateral and upper visual field. And again, yeah.
Can you remind me how old the kids are?
So our
Think
kids,
about reading.
yeah, so our kids here are between the ages of 5 and 12. So all of them can read somewhat, and there's a variability in their reading ability. And feel free to ask me questions. I'm very happy to take your questions. So if there's anything unclear or you have a question, feel free to stop me. So our kids are between the age of 5 and 12, and our adults are young adults between 22 to 27. Okay, so if we look again on the shift of the center of mass of the visual field coverage, you can see that in the dominant hemisphere for reading, which is the left hemisphere, we get an increase in the foveal bias. And actually in the right hemisphere, we actually have a repulsion from the center, whereas the visual field becomes actually shifted further away. The visual field coverage gets shifted further away from the fovea. So we see this really interesting development that we get this asymmetric, hemispheric asymmetric development that's tied to category, where we see an increase in the foveal bias in the dominant hemisphere. So it's right hemisphere for faces and left hemisphere for words. And on top of this, this is really nice because we see a protracted development of the visual field representation in these higher order areas during childhood development, even as the earlier retinotopic areas seem to be already adult-like in their properties. So we are really intrigued by this finding. And we were wondering whether there are any behavioral ramifications to this change in the visual field coverage across development. And basically, we thought there might be a link between the visual field coverage in the dominant hemisphere and the way people interact or look at faces and words. So adults typically, when you ask them to do a recognition task, this is the average fixation of 11 adults, adults tend to fixate on the center of the face. And we think this is a good strategy because it puts the computational resources of their right face selective region, which is kind of foveally biased and extend to the ipsilateral visual field, and where there are informative features for face recognition, for example, the internal features of the face. However, we hypothesize that this might not be the best strategy for children. And the reason is that if a child will fixate on a face, let's see where its visual field coverage will land in its right face selective region. So I'm just plopping over that face, the visual field coverage of the right p-fused faces, and you can see that the basically highest density of coverage is not actually on the informative features. So what we reason that the children should do is shift their gaze rightwards and upwards, because then it will put their computational resources in the face selective region on the informative features on recognition. So this makes a very strong prediction, is that there would be a link between the visual field coverage and looking behavior on faces and word. Of course, this is a prediction that can be experimentally tested. So basically what we did, again, during the experiment when we were doing the pRF mapping, the subjects are fixating, but here we're doing a behavioral experiment outside the scanner, and we're going to do a free viewing experiment. So this is our eye tracking lab. This is one of our children participants, and we're asking them to do a behavioral study. So in the behavioral study, it's a recognition memory study. They first have an encoding phase. They're going to see a stream of stimuli, including faces and many other objects. And occasionally a stimulus is going to repeat, and they have to press a button when that stimulus repeats. Then there is a 3-minute delay, and during that 3-minute delay, they see an irrelevant stimulus, whereas they see a rotating checkerboard, a black and white checkerboard on the screen. After that, they have a self-paced recognition test that they're free to move their eyes. So basically, in each trial, they see a stimulus, for example, this stimulus, and they have to decide if this is a stimulus that they've seen before during the encoding phase, or it's a new stimulus that they haven't seen. And during this free viewing, we're going to measure eye movements, because this is when they're attempting to do a recognition of that stimulus. And what we're interested in doing is comparing the fixation pattern in children to that of adults on the same stimuli. So this is an example fixation pattern that I showed you for adults on this face stimulus, and you can see that it's adults tend to fixate most on the center of the face. And on the same stimulus, you could see where children fixate. And you can see that on this stimulus, the children fixate rightward and upwards, basically consistent with our prediction that they're going to put their visual field coverage, they're going to put it towards their informative features are on the face. Now, this is just an example stimulus, and you want to quantify it across all the stimuli because they're somewhat jittered on the screen. So what we're going to do is for each of the stimuli in our recognition memory experiment, we have 16 different stimuli. We're going to put on that stimulus the average fixation pattern of adults, measure its center of mass, put on the same stimulus the average fixation pattern of children, calculate the center of mass, and then with a vector determine in what way children have biased viewing of that stimulus compared to adults. And we're going to do it separately for each stimulus, and we want to see if there's a consistent bias in the way that children look at faces and words compared to adults. So this is the results for all the face stimuli, and you can see that children tend to be biased looking rightward and upwards compared to adults, basically that they're pushing their resources of their right p-fuse faces upwards and rightwards towards the interesting features are. And interestingly, they never look more leftward and downwards in faces, which would actually shift their visual field coverage really off the face. So this is consistent with our prediction from the face area. So let's think about what's going to happen in the word area. So in the word area, we see the opposite effect, that it's more rightwards and lower visual field in the children compared to center in the adults. So in this case, they should shift their, if the visual field coverage is going to determine the fixation, they should shift it leftwards and upwards, compared to the adults. So first look at how adults look at words. So adults tend to fixate in the center of words, kind of like faces, which is consistent with their foveal bias. And on this example word, you can see that consistent with this prediction, children shift their fixation upwards and rightwards. And again, if you measure them across all the different words that we've shown, which are jittered and shown in somewhat different orientations, you can see that there's a consistent bias that the children fixate upwards and rightwards compared to adults. And again, they never are biased lower and, this is, I'm on the other side. This is upwards and leftward, and they never look downwards and rightwards, which would shift their visual field coverage further off the face. So we were really excited about this result. And the reason that we were really excited about this result, because it suggests a link between the computational resources and the ways it covers the visual field in your high-level visual areas, and basically how you view faces and words in the natural world. And this is also exciting because it suggests an important link between the development of the foveal bias in these higher order areas and natural viewing behavior of these stimuli. So this is one experiment that I wanted to show you. In another experiment, we wanted to test another prediction of this theory called eccentricity bias. And basically, and this question really goes back to the first question that I asked, is whether we can use eccentricity bias to predict where category representations emerge in VTC. So I started this talk by showing you this large-scale eccentricity bias in ventral temporal cortex. So now I'm zooming on the ventral view of the brain. So again, lateral regions have a foveal bias, medial regions have a peripheral bias. However, since Rafi's discovery, other large-scale representations of the visual field of the ventral stream have been reported. So another representation that also has been shown to show a lateral medial bias is this representation of animacy that's reported by several labs, including Jim Haxby labs, Alex Martin, Karamatsa, and Tyler Konkel's lab. And basically what they showed is that lateral aspect of VTC prefer animate stimuli, and medial aspect of VTC prefer inanimate stimuli. A third topography that also has this ventral lateral medial bias in VTC is this a topography of rectilinearity. So Roger Tuttle and Shane Nasser showed that lateral VTC likes curvy stimuli or responds more strongly to curvy stimuli, and medial VTC responds more strongly to linear stimuli or things that are less curvy. And we looked at this and we were actually really intrigued by these findings for several reasons. First of all of these representations have this lateral medial gradient. Second of all, they're really very anatomically consistent. So I've marked in all of these are different participants, by the way, because these are data from different studies. We marked a sulcus that bisects a fusiform gyrus, the MFS, into a lateral medial. And you can see that the gradient in all of these representations is really nicely aligned to the same anatomical structure. So we thought it's really cool because we have now a representational space that's actually anchored to a physical space in the brain, and that physical space is really anatomically predictable. So then we thought, oh, maybe if this is really a representational axis, maybe if I could measure how much a stimulus is foveal or peripheral, or animate versus inanimate, or curvy versus linear, I could predict where in this lateral medial axis that representation of that stimulus should emerge in ventral temporal cortex. So we started the exercise by looking at faces and places because we know that they have this lateral medial organization. So we measured their representation or their features along any of these axes. So it turns out that faces are more foveal than places. I already showed you this. They're also rated to be more animate than inanimate, than places. So places are rated to be inanimate and faces are rated to be animate. And if you measure, we use the rectilinearity toolbox provided by Saeed Nasser, faces tend to be curvier than places that tend to have more linear components. So this is kind of cool because all of these axes would predict that faces would be in lateral VTC and places would be in medial VTC, but you can't really dissociate. If you think about these as computing hypotheses of what governs the development of VTC, you can't really dissociate between hypotheses if you're going to keep looking at faces and places. So what you really need to do in order to dissociate between these hypotheses is find a stimulus that's heavily or intensely looked at and perceived in childhood and would generate very different topographies according to three different axes. So Jesse came up with this idea, and to be honest, initially was trepidation because he wasn't sure how I'm going to react. But it's based basically on his own experience. So Jesse was a child of the 90s, and in 1996, Nintendo invented this game called the Game Boy, and it had Pokémon stimuli. And basically, you're heavily rewarded for discriminating and recognizing hundreds of Pokémon. Now, given that this is the 90s, the graphics were very limited, which meant that Nintendo had to use monochromatic stimuli. They did not move. They're always shown in the same location in the screen. They were big pixels because, again, the graphics were a little bit crummy. And children actually held that at a pretty constant distance from them because it was governed by the length of their hands, right? So now you have perhaps this perfect stimulus that can dissociate your hypothesis because it would actually so you have to have actually two components of the hypothesis. One component is that if you see a stimulus that you experience intensely during childhood and have to discriminate, it would generate a category representation in VTC, so that has to happen. And second of all, maybe we could predict whether that category emerges depending on the visual characteristics of the stimulus. So let's measure where Pokemon land according to each of these axes. So because the Pokemons were small, they're actually more foveal than faces. So this eccentricity bias predicts that if a category representation will emerge for Pokemon, it should be more lateral to faces across this eccentricity axis. If you ask novice people, we get Stanford undergrads to rate how animate Pokemon are, so they actually rate them to be intermediate in animacy between faces and places. So that would predict that it would land kind of in between the representation of faces and places, somewhere in the middle fusiform. The third thing that we did is we measured, based on the visual characteristics, how linear or pixelated or curvy the Pokemon are. And because the pixels are really big, Pokemon are actually more rectilinear than places. So this axis of rectilinearity would predict that the emergent representation of Pokemon should be more medial than the place area, the most medial aspect of VTC. Okay? So what we need to do is run the Pokemon experiment, which was what we did. So this experiment is only in adults, and these are young adults that basically either had extensive experience with Pokemon when they're children, so they're between the ages of 5 to 8, they play hundreds of hours with their Game Boy, or adults that are similar in age but never played the game during childhood. And in order to verify that people have different knowledge of Pokemons, before participating in our scan, they had to do the Pokemon quiz. So in the Pokemon quiz, you get a picture of a Pokemon. It's basically the original Nintendo picture that's pixelated. You get 5 names, and you have to tell what is the name of that Pokemon. Everybody can identify Pikachu, even I can do that. But so some of our novices can know some of the names, but they're very different than their performance in our experienced participants, which can name basically almost all or all of our Pokemon stimuli. So once we have these 2 groups of participants, we're going to scan them in the Pokemon experiment. So since we're interested in the topography of Pokemons compared to faces and places, we're going to have these stimuli in our study. You might want to control for other things like animacy, because that's another axis that we're interested in, and maybe other cartoons that people had seen when they were children. And in addition, we're going to use other stimuli that we tend to use in our category localizer experiment, such as bodies, cars, or words. So the first thing that we want to do is to look at the ventral temporal cortex of both an experienced and a novice participant to just see if they have the general same topography. So this is an example, right, a ventral temporal cortex of a novice participant, and this is an adult participant, an experienced participant. They're both adults. And basically, you can see that they have the standard topography, that face representation or face selective voxels, shown here in red, are in lateral VTC in both our novice and in our experienced participants, and place selective voxels are in medial VTC. And I'm showing you Zan's thresholded map so you can see the whole picture. So then the question is, what happens is a representation of Pokemon stimuli in our brain. Okay, so this is our novice participant, and this is a topography of the Pokemon map, and not much, to be honest. People that don't have a lot of experience with Pokemon don't really show any voxels that show preferential selectivity or negative selectivity to Pokemon. However, the story is very different in our experienced participants, where you can find that there is a clear topography for Pokemon stimuli, and it looks like it's mostly in the lateral ventral temporal cortex. So this is the first part of the hypothesis, that it looks like experience during childhood can generate a new category representation. So in order to test our developmental hypothesis, what we're going to do is quantify the level of selectivity along this lateral medial gradient of VTC. So we just anatomically partitioned the VTC to 4 anatomical compartments, the most lateral one on the occipital temporal sulcus, then the lateral fusiform, the medial aspect of the fusiform, and then the collateral sulcus. And in each compartment, we're going to measure the selectivity to Pokemon, the selectivity independently to faces, and the selectivity to places, and see how this gradient of selectivity varies across this lateral medial axis. I'm going to show you data from our experienced participants. So each line here is a participant who is experienced with Pokemon, and this shows you across the VTC this gradient of Pokémon selectivity, where you can see that the peak Pokémon selectivity tends to be on the most lateral aspect of VTC. And you can see that this is more lateral than the selectivity profile to faces, which tends to be on the lateral fusiform. And it's definitely more lateral than the selectivity profile for places that tends to peak in its selectivity in the most medial compartment of VTC. So we thought this is really very cool because this shows that this kind of profile of selectivity is really consistent with the eccentricity bias hypothesis that predicted that because the Pokemon were foveal, we should see them emerging in the most lateral aspect of VTC. So our results suggest that a consistent viewing experience during childhood leads to the formation of long-lasting categorical representations in adults, and it's on the eccentricity that stimulus is most often seen with. So I would like to conclude and say that our results suggest that viewing experience and eccentricity bias are important factors that constrain the functional development of representation in human ventral temporal cortex during childhood. This also has important implications because a flip side of our finding is that a typical viewing experience during childhood might have profound effect on nascent cortical representations. Now, before I finish, I would like to state that I've shown you today only functional factors that affect the development of ventral temporal cortex, but we do not think that these are the only factors that might shape your representation. And other factors such as white matter connections, the myelo and cytoarchitecture might also constrain the functional development of ventral temporal cortex. And in fact, other research in my lab actually actively looks at this kind of anatomical development. So this is a study that we've published maybe 6 months ago. And basically in this study, we actually wanted to see if there are any anatomical developments in these high-level areas that shows this protected functional development like face and word selective regions. And we see that both the T1 relaxation time decreases as well as the mean diffusivity decreases from childhood to adulthood. And this decrease is associated with tissue growth. So we actually see anatomical changes in the same regions that we see this functional development. And we're doing these experiments in collaboration with other labs, trying to look at the underlying histology, trying to understand actually which tissue compartments are changing as your function is being developing during childhood. So with that in mind, I would like to thank you all for your attention, and I'm open for questions. I don't know.
All right, great talk, Patty. So the eccentricity story is essentially a retinotopic story. How do you contrast that with, the Kunkel and Karamatsa story that is actually world size in the world that's represented, say, laterally as small and medially as big?
Yeah. So there's a large coupling between real world size and the eccentricity bias. And as I stated in the beginning, scenes are also physically big. So this is basically no matter where you're going to fixate, they're always going to extend in the periphery. So Pokemon stimuli are actually smaller than faces. So of course faces are, depending on your distance, the size of the face is going to vary a lot. But if you're really far away, Alice O'Toole has these experiments that shows that people actually don't recognize faces when they're really far away, because like if you have a 1 degree face, you actually don't have enough visual acuity to recognize a face. So even people think that they recognize people far away from the faces, probably not. It's probably about their bodies, their gait. So I'd say that a lot of the faces, and we've looked at this, there's kind of like, there's a comfortable, like I see, talking distance that's maybe 1 meter or 2 meters away, that this is probably like where you see a lot of the faces. So, and again, when you're going to do recognition, you really have to fixate on the face. So this foveal vision is really about being able to use your high acuity to discriminate among faces. And so I really think it's really about your foveal vision more than the absolute size of the object in the world. Yes.
Very nice talk. This, sort of finding these connections and biases that can lead to the development of faces reliable in a specific area or places in a specifically reliable area. There's a lot of great evidence for it. It kind of makes a nice sort of intuitive sense. But there was a paper that came out, looking in congenitally blind individuals and finding with auditory stimuli that you get, these faces and places have this similar organization. And then recently, doing this with haptic recognition of faces, feeling faces and showing it there. And that seems to really challenge the idea of, why would faces pop up in the exact same place in a totally non-visual domain?
So I feel that these studies are really good at framing some ideas in your head that's not exactly the evidence in the study. So I'm kind of being a little bit blunt about it. So basically, if you're referring to a study by what's his last name, and Hans Utebeek, which was published several years ago in PNAS. In that study, they had auditory categories presented to blind people and sighted people, and for the sighted people, they also showed the same categories visually. And what they found that if you look at if you try to decode the category of the object, in the sighted people, we know we can do it from visual stimuli. In the blind people, you could get this same kind of categorization from auditory stimulus, and you could use the visual stimuli to predict the blind people's data and auditory. You couldn't do it, so that is basically the evidence that you don't really need visual experience in order to shape these category selective vision. But if you look at their data more carefully, it never quite shows that the representation in blind people is equal to the representation of sighted people. In fact, you cannot decode from auditory stimuli in the blind the visual stimuli in the sighted. Second of all, the performance in the blind from auditory to visual is not at all as a visual to visual from different sighted people. So, and it's really big effect. It's not like they're not even trying to hide it in any particular way. And the third of all, if you take a sighted person and shows them the same auditory categories, you can decode in a sighted person the category of the object from auditory stimulus. So this is just to make sure that you guys, before I answer your questions, just remember what is that evidence. So I think that study shows that it's not necessary to form category representation, but it really shows that you get the same representation in blind people and sighted people. And this is kind of what that study shows. Where I stand in this debate, I think that there are some things that are probably already there at birth. So what is probably there at birth is like the pattern of cortical folding. So you don't sprout a sulcus or a gyrus after you're born. The long-range connections are probably there, so the big fascicles are probably there, and they constrain a lot of the organization. But you do need the visual experience in order to get the typical adult-like experience. So there's something that's going to be common in the sighted and blind people. It's going to be these anatomical scaffolding. There are also aspects of our experience that are going to be common. But you really need this visual experience to get a typical ventral temporal cortex that you would have in a typical sighted adult.
Do you mean like high-level connections? Because presumably it wouldn't be auditory connections present at birth diffusiform gyrus that are causing face.
There is a fascicle that's called the arcuate fascicle, and it goes from the fusiform, it arcs through the STS, and it goes to the prefrontal cortex. So people have claimed that there are feedback connections from auditory cortex to visual. Definitely there are connections from prefrontal cortex to visual cortex. So in the absence of bottom-up visual experience, you still have the top-down visual experience. So I think these connections are already present at birth. Yes?
Thanks, that was great. So I was really intrigued by the similarity in the developmental trajectory for the mobile bias for faces and words.
Yeah.
But then one thing that seems a little bit interesting about the story is that it has to do with the experience of just like where you end up seeing, faces and words, in your normal visual experience. And I was thinking, especially for young children, the amount of despite similar developmental trajectory, the input seems to be really different for faces and words. And it seems like you would get tons of input for faces quite before 5, words maybe starting around 3 or maybe 2. And then I was thinking, well, maybe you could use some of your, I wonder if you had, so you were saying you had variance in the 5 to 12-year-olds in their verbal or their linguistic abilities. If you could use that as sort of a measure of the extent to which they've been exposed to words, and then ask whether or not the linguistic abilities actually predict the foveal bias, because that would be evidence for your kind of like, experience base.
So we didn't do this in this particular study because we don't have enough people to do an individual difference study. So the behavioral data is only in a subset of our participants, so this is not enough to actually do a statistically significant correlation at looking at individual differences. We are scanning these children longitudinally, so eventually we'll have this data. We have another study that we actually looked at the development of the representations of faces and words and all these 10 stimuli categories, just looking at the distributed responses. And there we can actually see that there's the distributed representation of words really develops very profoundly in this age group. It's actually anatomically specific to the lateral VTC. So we have, and it's in the left side. So it's really selective for words and not for numbers. So the number representation doesn't develop. And this is actually coupled with their reading accuracy. So we can see that there is a relationship between functional development and reading ability, but for this study, we just don't have a big enough sample size to look at individual differences at this point in time.
So are you surprised that it was a similar developmental trajectory? Like, would you not have predicted that you would have seen this foveal bias for faces, develop earlier than you would have seen for words? Or?
There is a little bit, so I don't have enough data to be confident when it starts, but where's this picture where I have both hemisphere? Sorry. I'm looking for a specific picture, just bear with me. Here. So it does look like the foveal bias in the faces already is a little bit higher, but again, I haven't measured this earlier, so I really don't know if it's been like that forever, or is this because you have more experience with faces. So I think this is something that we still have to look at. It's really hard to put younger children in the scanner because we really need them not to move, and 5-year-olds are already a challenge. Yes, maybe Carolyn and then Brad.
I'm just curious what your predictions would be for other high-level visual categories, like, say, scenes, and the peripheral preference for those regions of the brain. Do you expect not to see a development in age if you look up and see selective areas, just because children are not necessarily training on scene perception tasks they're developing as much as on learning to read?
So what we see with other properties, like looking at selectivity, like how many voxels are selective to category, or if we look at information and distributed responses, it does seem that face and word have a particularly protracted development. So for example, with face selectivity, we still see development in adolescence compared to children. With place selectivity, it kind of comes and goes. So in adolescence, we're sure that it's adult-like, is it like the PPA and also the information and distributed responses, but in children, in young children, it kind of varies by hemisphere. So we see a little bit of development, but it's not as strong. So, and if we look at the tissue changes, this is the one I showed you at the end, we really don't see profound tissue growth that we see in the place selective regions like we see in the face selective region. So my working hypothesis is something about these regions are particularly taking very long time to develop, and this region develops earlier. And this could be because of actual our life experience. So one of the reasons, this is kind of like a hand-wavy explanation to some extent, but it's really fascinating to me why face recognition still improves. So word reading is kind of obvious, like you don't learn to read before you go to school. So this is like an ideal age to look at. But what about face recognition? And I really think that the size of your social group really increases a lot during development. So maybe when you're a kid, like how many people you need to recognize. You have a few friends in school and like mom and dad and grandpa, but mom and dad and grandpa and your brother really look very different. I mean, it's like there's so many cues that are going to differentiate them, right? But once you go, let's say, to middle school and high school, and even college, your social circle really expands, and you actually have to discriminate between a lot of people that are about the same age. So it really presses the demands on remembering more faces and really doing fine-grained discrimination, that I think this is why this development is particularly protracted. So back to your question, because I haven't answered it, based on this logic, I would think that a lot of this is much earlier developed than in face-selective regions. We have the data, we haven't looked at it, so maybe in a couple of years I'll be able to tell you the answer to the question.
I
Brad.
actually didn't have a question, but I just want to follow up on what Jonathan said.
Yes.
Before I had a child, I used to think that through word exposure in kids, it started pretty late. But having seen my daughter, how much exposure she had to words, it starts pretty early. They're looking at a lot of words.
Written words.
Yeah, so I don't think it's as distinct from faces as I used to think. I think face exposure is great. What's that?
You think they're phobiating a lot of words compared to having a book held
I
up in
think
front of
they're
your
looking at
face?
books a lot. I mean, undoubtedly they're getting more experience with faces early on. I don't think it's as different as it used to be.
So one hypothesis that's been put out by Marlene Berman and Stanley Han is, why does this lateralization come about? So one of the things that we've seen, so for example, if you look at face-selective regions, are not lateralized in children, but they become more lateralized across development because of face selectivity really increases more in the right hemisphere than the left hemisphere, and the same is but for the other hemisphere is for reading, for words. And both Stanley and they have different variants of this hypothesis, and Marlene Berman thinks that this lateralization has to do because of competition of foveal resources. So because both words, reading, and face recognition require you to foveate on the stimulus, and the word or the language network is really left lateralized, kind of words win the left hemisphere, so faces get pushed to the right hemisphere. I don't know if that's true, and again, this is something to really look at in a longitudinal study to see if that's true, but that's one of the working hypotheses of why this lateralization may form during development. Yeah?
This question just builds on your answer to the last question.
Oh.
I thought that the hypothesis about the increasing social network size of children was really interesting. It reminded me to ask, how do these results compare to the monkey deprivation work that's coming out of the Livingston lab? I imagine that monkeys in that lab setting are not increasing in their social network size over development. Does the foveal bias of FFA in those monkeys increase over time?
So for those of you who don't know what the studies that Carolyn is referring to, Michael Arcaro and Marsh Livingston had a couple of years ago a really interesting study. They raised monkeys, so they have a lab with monkeys, and they have newborn monkeys, and some of the newborn monkeys, I don't remember if it's 3 or 6, they raise them without seeing faces. So basically, one of the monkeys, they raised a monkey and they came up with these like welder hats, so the monkey never saw their mom's face or anybody else's face. The other monkeys were raised by their mom, but the mom's got like a Darth Vader mask. So really, so basically they couldn't see the face. Sometimes she told me that sometimes the monkey cheated a little bit and they tried to lick a little bit, so maybe saw a little bit of the mouth. But basically these are monkeys that get visual exposure but not the faces. And basically what's really interesting is that they scan these monkeys across the development. I think monkeys become kind of teenagers when they're 2 and adult when they're 4. So this is a much faster time course. And basically and then they have like regularly developing monkeys that they're measuring at the same time point. So regularly developing monkeys take some time for the face patches to emerge, and this is around day 200 of the monkey's life. But the face-deprived monkeys don't show face patches, even though they show other selectivities like selectivities to the hands or limbs or selectivities to places. So basically this is a really strong evidence showing that experience to faces is necessary basically to the formation of face patches. So Carolyn asked me how do my study relate to their study. So it relates because it shows that experience plays a role in shaping these regions and getting the adult-like selectivity. I cannot answer with my studies this question, is face recognition innate or not, because my youngest child has 5 years of experience with faces, which you should say is really a lot. So, I mean, of course, you would like to do this experiment in infants. Rebecca Zach's lab has scanned infants. They scanned 15 hours of data on infants and got 15 minutes of results, because it's really hard to tell the baby, please don't move during my fMRI scan. But they claim that there is this like face-place separation already in around 6 months old. But then you could say, well, 6 months old still is 6 months of experience. So I think that the evidence suggests that experience is really important for the formation of these regions, and this is partly my answer to Sarah about how it relates to like the Karmatza and B results. But there might be other factors that also scaffold this ventral stream. So people are not like really born to below access, so they have an anatomy that's already set. Okay.
I have at least one question, maybe more I can follow up on later, I guess. So you made the case, I think, that it's this eccentricity bias that's really driving the results in the Grill-Spector 2019 paper. But does that argue against those other explanations? Because Pokémon are also animate and curvy.
So basically, I think it does. And the reason is because if you take these axes as face value, if you measure where the Pokémon lie along the animacy axis, it would predict that they would emerge, the representation would emerge between faces and corridors. And we also actually measured, because we had animal in the stimuli, the topography of animals. And animals have a topography that's both, so you really see the stretch that's in the middle, and you see this additional patch on the lateral side, and that's consistent with Kunkel's result. So we do see a little bit covered with some animacy, but we really don't see where the peak selectivity of animacy should be in medial VTC. So we think that, according to this predictions, this representation should have emerged elsewhere on this lateral medial axis, and it's not there. If we look at curvilinearity, you really should have had, these are actually rated to be these are measured to be much more linear than the places, and then it would have predicted that this representation should be on the medial aspect of VTC. So I just showed you like one participant, but we also have a group map, and it really looks like that. You really see stuff in the lateral VTC, and you don't really see stuff in the medial or in the middle of the VTC or the most medial portion of the VTC.
Okay, so I guess my question is about really how you assign the linearity. Is it because of those stimuli?
Yeah, so we actually,
Game Boy that are very sort of straight?
Yeah, so basically this is a toolbox that basically measures how much curvy or linear, and it gets you an index. It's the same Nasser's toolbox. So, and it basically gives you, so we ran all our stimuli that were in the experiment through that toolbox and just measured how curvy or straight they are. We actually, Peter, you asked me about world size. We actually asked people to rate the size of Pokemon. So for those of you who are Pokemon experts, it turns out I did not know this. I learned a lot about Pokemon that I didn't want to know. But Pokemon actually on the game cards, they have sizes, they have powers, they have a bunch of stuff. So they actually have a physical size according to these cards. But we also asked like novices to rate these stimuli, and a lot of them kind of look a little bit like animate stimuli, so people will kind of give ratings to them. Size in the Pokemon is going to be very correlated. That's kind of partially an answer to the photo device. So we wouldn't predict them to be, let me see, maybe it's wrong. Size would predict them to be here. So they're basically rated in size, real world size. I don't know if Pokemon really have a real world size, but how people perceive them, had they seen a real world Pokemon, they're actually here. Because they're animate, the people usually rate them to be, on average, bigger than faces, but not as big as places. So actually, real world size is something like this, and this is not a topography that we got in this experiment. Yes?
So I have a question about the cortex that's responding to words or letters.
Yeah.
So, there are still people in the world who grew up without reading, and we didn't evolve to read because, 5,000 years ago, nobody was reading.
Yeah.
So if you found, you go to Papua New Guinea Highlands or something, you find these people, what is that cortex responding to?
That's really interesting.
That's one question. The other thing is,
Yeah.
some word systems are really small, like Chinese letters are very compact, whereas, like Indian words are really long. Would you predict that Chinese would be more laterally represented than Devanagari script?
I don't know. This is a really interesting question, and I don't know the answers because I haven't really studied people across cultures. I'm interested in this laterality because Hebrew rate goes from right to left, so I don't know how it's going to interact with this right versus left hemisphere symmetry. And I'm trying to convince my friend and colleague Michal Ben Shachar in Israel to do this experiment in children. So this is one. So I don't know. About literacy, I do have some answers. This is not from my research. This is from Stan Dehan research. So Stan Dehan has a paper published 10 years ago that he looked at people who were illiterate in Brazil, and I think he mentioned mostly they were in Brazil. I think Sheng He also looked at people in China, that some of them were illiterate and some of them learned to become literate in adulthood. And basically, he has this idea of kind of cortical recycling that basically what you're born with is some region that has this foveal bias, and they get kind of recycled to do the task that's important for your daily livelihood. So basically, in these people that are illiterate, the same region, like you get a group map of where the word form era is, he thinks that they respond to faces. And so basically, he thinks that that kind of changes at the same territory. That's part of his hypothesis why you're fighting on the foveal bias. He has a study, his wife has a study, her name is Ghislaine Dehan, and she has a study that was published in PLS maybe a year or two ago, and basically she measured, so in France, children learned to read during first grade, so she scanned this 6-year-old like 6 times during first grade, and basically she didn't show what they were selective to be beforehand, but she showed that this region becomes more word selective as they become better readers during the first year of like reading instruction. And there's going to be some variability. Maybe some French kids also learn to know how to read before age 6, but that's basically the result. Some of the data in my lab suggests that that may or may not be true, and I might tell you this once we figure this out. But I think there might be actually more representation of body parts that gets recycled for reading. So, yeah. Okay, I think that's all for now.
Okay, thank you.
Thank you for your attention.